Today's lecture is going to step you through what you need to know about protein synthesis, okay? And proteins are really, really important. They end up making up tons of structures in our body. They're involved in like all the processes in our body. And if a protein is made improperly, or made not at all, it's going to lead to some serious issues, maybe some genetic diseases or disorders. Oh, hey, which we might be learning about soon. So we're going to get into the specifics of how proteins get made. Okay, so your cell isn't just going to make one for no reason. It's going to make one because it's needed, right? Some kind of message is received that, hey, we need this thing. Let's get going on making it. Now, as you know, DNA is the genetic code. So it's got all of that instructions as to how to make the proteins, but DNA can't actually make the proteins itself, okay? And as it turns out, in order to take DNA and make it into a protein, you first got to make some RNA. So making a protein is kind of broken down into two steps. The first step is called transcription, and this is taking a piece of DNA and making a piece of RNA that matches it. And then in the second half of protein synthesis, we take that piece of RNA and we are now going to turn it into an actual protein. Okay, so as you can see, RNA is a really important thing, and so we're going to learn a little bit about RNA and how that works, okay? You guys are already experts on DNA, so obviously don't feel the need to copy down the DNA stuff. You already know that, okay? There are a couple of things you need to know about RNA that make it different from DNA. The first is that RNA is typically single-stranded, okay? Instead of deoxyribose, the sugar is ribose. DNA is only found in the nucleus, and RNA is found in the nucleus, the cytoplasm, and also in ribosomes. And another main difference are the base pairing rules, okay? In DNA, we have A, T, C, and G. A goes with T, C goes with G, right? In RNA, we have 4 bases, but they're slightly different. We still have C and G, and they still go together. We still have A, but instead of A going with T, A goes with U. U stands for uracil, okay? So URACIL, uracil. And so those are the main differences, at least as far as what you need to know for the differences between DNA and RNA, okay? But of course, they're both genetic material. They both pass along hereditary info. They're made of the building blocks called nucleotides, and they both have like a 3' and a 5' end, okay? In fact, some things, like there are certain viruses where they don't even have any DNA, their genetic info was only RNA, okay? There are 3 types of RNA that we're going to be talking about today. There's mRNA, the little M stands for messenger, and that is going to be our little copy of a piece of DNA gene right here. Boom, mRNA. Okay, so we're going to make that from a template of DNA. There's also ribosomal RNA or rRNA, and that literally helps make up the ribosome. And again, the ribosome is an organelle that makes proteins. And then there's tRNA, transfer RNA, and this is going to bring an amino acid to the ribosome, okay? And more on this in a second, just like showing you these words so when they come up later, it's not a complete shock to you. All right, so like I said, protein synthesis is divided into 2 main parts, transcription and translation. So let's talk about transcription. Transcription is making mRNA from DNA. So let's get into some specifics here. You might have heard of a gene, okay? You're in a genetics class, right? Based on the word gene. Okay. So you know you have the DNA double helix, okay? And maybe you know that that our DNA can be coiled and condensed so that it forms a thing called a chromosome, okay? Well, what a gene is just a small segment of DNA. So there's a whole bunch of genes on this one chromosome. And as it turns out, a gene contains the codes to make one protein, okay? So one gene makes one protein, great. And a specific gene is going to be like turned on because again, your cell gets a message and the protein is needed. So that specific gene is now all of a sudden going to become very important and is going to be used. And this is going to happen in the nucleus. Why would this be happening in the nucleus? Where is DNA located? The nucleus, right? So this is going to be happening in the nucleus in eukaryotic cells, right? So here we go. What's going to happen, and this is going to look similar to DNA synthesis, is we're going to get a small part of our DNA to unwind, to unzip, unzipped. You're going to have a different enzyme instead of DNA polymerase. So when we were making DNA, now we've got RNA polymerase because it's going to make RNA. okay? And it's going to read the DNA from the 3' to 5' direction, just like in DNA synthesis, and it's going to build a complementary strand in 5' to 3'. And so what's going to happen is your DNA is going to separate, it's going to unzip, and a complementary RNA strand is going to form. Complementary because we're going to do base pairing. Here's a T, boom, an A is right there. Here's an A. Remember, there's no T's in RNA, so a U is going to go there. C goes with G, T goes with A goes with U, G goes with C, A goes with U, and so on, until we have a piece of RNA that is the length of our gene, okay? So I want you to try now, transcribe this segment of DNA into its complementary mRNA strand. Pause the video, jot it down in your notebook, and when you're ready, push play. All right, so hopefully you did it right, and here's your complementary strand. Remember, the 5' and 3' always go together. It's like heads and tails because things are anti-parallel, okay? So if this is 3' over here, there, automatically 5' is going to be here, and then we follow our base pairing rules, making sure that there are no T's in your mRNA strand. Anywhere you would normally put a T, a U goes there, okay? So that's transcription. Again, we took some DNA, and we just made a little kind of like buddy of RNA that pairs up with it. And notice it's just on the one strand. We just need one strand of RNA. So one strand of DNA is just going to hang out and chill. Okay? So that's transcription, making our mRNA from DNA. So now we've got our piece of mRNA. Cool. What happens next? Well, the mRNA is going to leave the nucleus through a little pore. Okay? Your nucleus has the little holes in it called pores, just like how your skin has pores. And it's going to go to the cytoplasm. Why? Well, as it turns out, you need a ribosome to make a protein. A ribosome is an organelle that makes proteins. And the ribosomes are in the cytoplasm. So your mRNA has to leave the nucleus and travel to the cytoplasm and find itself a ribosome. All right, so let's go back, right? First slide, protein synthesis here. We got a piece of DNA. It gets unzipped. Now we make a piece of mRNA. Here's our lovely piece of mRNA. It's going to come out of a pore in the nucleus, and now it's going to go and it's going to get translated. Okay, so we've done DNA to mRNA, we've done transcription, and next we're going to do translation. All right, so again, translation are our ribosomes making proteins using our piece of mRNA. Translation can be broken down into 3 parts: initiation, elongation, and termination. And we're going to go through each one of these parts. Initiation is going to sound just like, it's going to do exactly what it sounds like. Initiation means to start, okay? So basically, the mRNA is going to go near the ribosome, okay? And the ribosome is going to find the start codon. The start codon is always AUG. So the ribosome is going to be able to recognize AUG, and it's going to latch on. The ribosome comes in 2 parts, okay? So one little part of the ribosome finds the start codon, and then the other big, huge honkin' part is going to then come in and snap on top of it, okay? So the small little guy's like, hey, all right, we got some mRNA over here. And the big guy comes and whoom, whomps on, okay? And now it's ready to do something. So initiation is the mRNA meeting up with the little part of the ribosome and then bringing the big part of the ribosome in on top. Next is elongation, okay? This is going to be actually kind of building our long chain of amino acids. So our mRNA is going to get read by the ribosome in triplets, okay? In a triplet meaning a group of 3, right? And the group of 3 is going to be 3 letters on the mRNA. So maybe if our mRNA reads GCA, this would be the triplet that it's read in. This triplet is called a codon, okay? So if our mRNA reads GCA, then a tRNA, a transfer RNA, and we'll remember back here, there's 3 types, right? A transfer RNA, one of these guys, okay, that's our transfer, is going to bring an amino acid in. Which one is it going to bring? Well, it depends on the codon. So this is GCA, and I can use a chart that looks like this to help me figure it out. So my first letter is G, so I'm down here. My second letter is C, so now I'm in GC, so I'm right here. And my third letter is A. And as you can see in this whole square, it doesn't even matter, right? All of these code for ALA. That amino acid is alanine. Cool. So that tRNA is going to bring an alanine. Okay, so let's look at tRNAs here. The T stands for transfer. At the top, each one has its own different amino acid. At the bottom, it has an anticodon that's going to match up with the codon, right? So if the codon reads AUG, the anticodon is going to be the complement of that, UAC. Okay, cool. So this tRNA would bring MET. Okay, how do I know I can use my chart? But notice this chart is what the mRNA sequence is. So if the mRNA sequence, if the mRNA codon is AUG, I can go back to my chart. A, second letter is U, third letter is G, MET. And as it turns out, MET is short for a methionine, and that is basically what I think almost all proteins start with is MET. So AUG, that's why it's called the start codon AUG, because they all start with the same amino acid. Okay, so this RNA, it's got an amino acid on top, got the anticodon on bottom. These match up, so we know we're bringing the right thing. All right, so here we go. We got our MET. We've got our anticodon. This crazy squiggly thing is a tRNA. Great. So now we've got one tRNA in there with one amino acid. Okay. And what's going to happen is another tRNA is going to come and it's going to attach to this one right here, codon 2. Okay. And so it's going to line up with this one, and this one tells me that this is going to bring an arginine in. So the next amino acid to come would be arginine. I think this next demonstration does it better. So here's our first one, MET. Okay, then the next amino acid would come in, UUU, so the tRNA would have AAA on it. And as it turns out, that would bring the amino acid with the abbreviation of PHE, which is like phenylalanine or something. Okay, and then the ribosome, which is this orange and blue thing, is just going to like chunk down the mRNA to the right. And so notice as it does, okay, the MET is now going to get attached to the PHE. And so here's our MET attached to the PHE that gets strung together. Now this tRNA is totally empty, and it's going to leave, it's going to fly away, and it's going to go pick up another MET. Great. And so now the ribosome moves on down. So the next one's going to be CUG. So this one is going to come in. Okay, the ribosome is going to chunk on down the mRNA. And now we have MET, PHE, and now the PHE is going to get attached to the LEU. And so on and so on. And so the ribosome is just going to keep moving down the mRNA, and tRNA is going to keep coming and bringing new amino acids to elongate our chain of amino acids. Here's just another visual, okay, showing you this growing chain that's going to become a protein. And so here's a new tRNA coming. And then this PHE will get attached to here, which is attached to here, which is attached to here, and it's just going to keep on growing and growing and growing, or elongating. So the process of elongation are TNAs matching up with the codons on mRNAs and bringing amino acids and making a long chain of amino acids. Next, we get to termination, the terminator, right, aka the end. And so what's going to happen is our ribosome is going to reach a stop codon. There are a couple of different stop codons. And on the chart, you'll notice like, boom, here they are, stop, stop, stop. Okay, so the stop codon doesn't actually code for an amino acid at all. It just tells the ribosome, okay, it's time to stop. And so what's going to happen when your ribosome gets there is it's going to pop open back to its two original parts. And then now we've got our little chain here of amino acids, and this is going to fold up and become a protein, a 3D protein, because, you know, bodies are in 3D. So just like one more time for translation, initiation is where the mRNA meets up with the ribosome, little part joins and big part joins. Elongation are tRNAs bringing amino acids in, matching up with the codons, and we get a growing chain of amino acids. And then termination is pop, the ribosome comes apart. It releases our brand new protein that's made. Okay? And so this ends up being really, really important in biology. It's one of the main founding tenets that you go from DNA to RNA to make proteins. Okay, super big deal. And again, to get from DNA to RNA, that's called transcription. And to go from RNA to proteins is called translation. If you want to see more on this, there are some really great YouTube videos. If you just look on YouTube and look up like protein synthesis, I know Amoeba Sisters has a really good video. So if you're wanting to see like an animation, just put it into YouTube and you almost can't go wrong. There's lots of really good ones. If you kind of want to like see this in action yourself, I would recommend you go ahead and check those out.